Well, hi everybody. Today we are very pleased because we have Dr. Westholm that's going to give a presentation about his latest paper. Basically, Dr. Westholm obtained his PhD and MD degrees from the University of Lund. He completed his PhD thesis at Dr. Lena's Aliason islet cell exocytosis group at Lund University too. His PhD project was focuses on investigating the underlying mechanisms that diabetes-associated beta and alpha cell dysfunction. He's currently working as an MD in emergency and internal medicine, and today he will present the results. That, as I said, this is a very nice paper that he just recently published, and he is going to talk to us about that results. So you can take over.
Thank you very much for the introduction, Osvaldo. Thank you for the invitation to give this lecture. I'm very thankful to be in contact with you through my lovely fiance, Sofia Sifo. She's sitting here next to me. Today I'll be talking to you about my main project for my PhD, which was on the protein insulin-like growth factor binding protein 7, IGFBP7. And the title of my talk is IGFBP7, a potential new player in pancreatic islet crosstalk. I'll talk a little bit about the background of diabetes and the rationale for our studies, and then I'll go through the paper result for result, and I'll also take some of the results and put them into further and future perspectives, and also some clinical applications for measuring IGFBP7 that is in use already today. So let me get started. So I'll very quickly, let's see. I'll get us started with the basics. Type 2 diabetes mellitus, a disease I think all of us know quite a lot about, but I think it's important that we just set the foundations here for what we're looking at. Type 2 diabetes is, as you know, defined by elevated blood glucose levels, which can be due to several different causes. And the way we in research and also in the clinical setting, I can say from working in internal medicine, the way we view the disease, it is the result of two parallel pathogenic processes. We have, I'm going to see if I can get my marker here. Laser pointer. We have the two processes of insulin resistance in tissue such as fat tissue, liver tissue, muscle tissue, being regulated by several genes themselves that accumulates over the years, insulin resistance. And then we, of course, have insulin secretion from the pancreatic islets themselves. And what we know is that typically in type 2 diabetes, we have an initial rise in insulin resistance in these tissues. And that is true for most cases, but there are, of course, also cases in the clinic where we see, even though it's not autoimmune diabetes, where we have beta cell destruction leading to impaired insulin secretion, we do see cases where an initial impaired insulin secretion also is the first step in the development of this disease. But typically we see elevated insulin resistance over time, and then a response from the beta cells with increased insulin secretion, and then at a certain level the beta cells fail and we have an inappropriate or inadequate amount of insulin secretion for the amount of insulin resistance needed to overcome insulin signaling in target tissue. So that's what leads to elevated blood glucose levels, and elevated blood glucose levels leads to all the type 2 diabetic complications that we so dearly want to avoid. Pancreatic islets, the organ that I studied the most. These are highly vascularized endocrine microorgans within the pancreas. They constitute around 1 to 2% of the entire pancreatic weight. And we have in humans, beta cells constitute roughly half of the number of cells in pancreatic islets, and then we have the alpha cells, about a third to a quarter of the cells, and then we have the PP secreting cells, pancreatic polypeptide cells, the delta cell secreting somatostatin, which is like a general inhibitor of both beta cells and alpha cells, and we have the epsilon cells secreting ghrelin. And the balance of how these hormones are secreted throughout a day in response to several environmental triggers such as stress, food intake, most importantly, regulates our entire metabolism. And these are in turn, of course, also regulated by other circulating hormones, both from the HPA axis and from the gut. If we zoom in even further, we find the beta cell, my favorite cell type. And as I mentioned earlier, dysfunctional insulin secretion is a key step in the development of diabetes mellitus. And since I'm not sure how well all of you in this meeting know about the stimulus secretion coupling pathway in beta cells, I will go through it now. If you already know it, I'm sorry if this is old stuff to you, but if you don't know it, this is quite important to understand the talk I'm about to give. So please bear with me. We have in the beta cell, we have the primary transporter for glucose being the GLUT1 transporter, taking up glucose from the bloodstream, which is then metabolized via glycolysis, generating pyruvate for the TCA cycle. This leads, of course, to an increased ATP to ADP ratio in the cytosol, which closes the potassium channel or the KATP channel, the potassium, the ATP dependent potassium channel. And this closing or decreased open probability of the potassium channel leads to successive depolarizations of the plasma membrane with the subsequent influx of first sodium and then calcium, and influx of calcium through voltage-gated calcium channels, L-type channels being the most important ones, and also the concentration of L-type channels near the insulin granules is the triggering step for insulin secretion in beta cells. And pathogenic processes resulting in impaired insulin secretion from beta cells can include a multitude of pathways affecting all of these steps. It could be perturbed mitochondrial function, it could be misfolded vesicular proteins, and it could also be the way the cells talk with one another, altered autocrine and paracrine crosstalk. So where does IGFBP7 come into all of this? IGFBP7, as I said earlier, stands for insulin-like growth factor binding protein 7. And this protein has already been described to be involved in cardiometabolic diseases before. It's elevated in the serum of men with newly diagnosed type 2 diabetes. This was shown 20 years ago in this diabetes publication. In a Nature Metabolism publication from 6 years ago, we have that local production of IGFBP7 from Kupffer cells or resident liver macrophages can promote lipogenesis in hepatocytes. So it seems to be a sensitizer of insulin secretion in hepatic tissue. Quite interesting. And also this paper here in a mouse heart failure model showed that IGFBP7 could promote the development of fibrotic tissue in the heart. And this is actually a really good paper. I highly recommend people interested in cardiology and metabolism to read this, if you like my paper, because they show this in every conceivable way possible that IGFBP7 is related to heart failure here. And interestingly enough, IGFBP7 is also implicated in reducing insulin secretion in a ferret model of cystic fibrosis-related diabetes. So we do already have some data indicating that IGFBP7 is involved in islet function. So what is this protein? Let's look at it at a more closer biochemical level. In the IGFBP family, we have the IGFBP126 proteins, which binds the IGF-1 hormone and regulates the bioavailability of this hormone in blood and peripheral tissues. Here, IGFBP1 and 3 are the main members of this family that have been studied the most, whereas IGFBP7 has been shown to interact with receptors for IGF-1 and insulin. But more importantly, IGFBP7 binds to insulin with a greater affinity than to IGF-1. So this, we see just an example of this interaction here with IGFBP7 in yellow and insulin in purple here. And the binding affinity for insulin for IGFBP7 to insulin is greater than that of IGF-1. So clearly we see that this protein is involved in a lot of interesting and related diseases, and it also has this very, very, very interesting biochemical property of binding to insulin and itself, and also modulating insulin receptor signaling. So with this in mind, we set off in my project here. We have the two aims, of course. The first one was to investigate on a purely descriptive level, investigate IGFBP7 expression in human islets in healthy donors and donors with type 2 diabetes. And then we also wanted to perform mechanistic studies and evaluate IGFBP7 effects on beta cell function. So let me just grab some water and continue. Our first results. We see that IGFBP7 as a whole is upregulated in the type 2 diabetic state. It's not this is whole islet RNA sequencing data from a total of, I think it is 217 human donors. And here we present it as normalized count data with the division for non-diabetic donors, donors with impaired glucose tolerance, and donors with type 2 diabetes. We do also see, when we just split it up by sex, that it is also more highly expressed in men than in women. Interestingly, when we look at the HbA1c values of these donors in the donors without type 2 diabetes, so so to kind of get a sense of how is the correlation before we say that the disease has developed, we don't see any correlation of the IGFBP7 mRNA counts and long-term blood glucose levels in non-diabetic donors. So there doesn't seem to be affected before you, or it doesn't seem to be affected in this state, period. And also when we look at all donors and IGFBP7 to age, we also don't see that it seems to be related to age at all. So here our sense is that it's simply connected to the disease state as such, rather to any classical risk factors for type 2 diabetes, age being the most important one here. We then wanted to look more closely at IGFBP7 in human islets themselves in experiments. So we had this workflow where we had human donors that where we obtained their pancreases, I'm not sure on the plural there, and digested them, picked islets and split islets for a 72-hour treatment with a blank BSA control, and then IGFBP7 dissolved in BSA, and then we performed a very simple experiment layout, as you can see, and then we performed endpoint analysis with glucose-stimulated insulin secretion, RNA-seq, and Western blot. So our initial results, here we have only islets from non-diabetic donors, from 6 non-diabetic donors. We see that in the high glucose stimulation after a 72-hour treatment with IGFBP7, we have reduced insulin secretion. We have reduced insulin secretion here. And so this is a very interesting first step. We have IGFBP7, which we know is can modulate insulin receptor signaling itself in beta cells, and when we add this to beta cells, we see a reduction in insulin secretion. This we then recapitulated, or in endo-C beta H1 cells, which is a human fetal beta cell line. We see the same results after a 72-hour incubation with IGFBP7, removing the medium and then just performing a blank glucose-stimulated insulin secretion on the cells. We see a reduction in glucose-stimulated insulin secretion after IGFBP7 treatment. However, we don't, when we perform these experiments, but stimulate using a high potassium containing solution, thus we offset the electrochemical gradient between the outside and the inside of the cell. So we kind of mimic the closure of the KATP channel. We don't see any difference in glucose-stimulated insulin secretion, and thus this kind of tells us that IGFBP7 works by affecting steps of the insulin secretory pathway, the stimulus secretion pathway, before the KATP channel. So either in glycolysis or in the mitochondrial metabolism. We also perform these experiments in acute treatment of the cells with IGFBP7. So we had IGFBP7 present in the stimulation medium, and there we also saw the same effect here. Acutely, the IGFBP7 also increases C-peptide secretion, because we measured in those days. IGFBP7 interfered with. I went on to these donors to understand what genes were affected by IGFBP7 treatment. And 2 genes fell out. We have PAC1, P21 activated kinase 1, and INPP5B, inositol polyphosphate 5-phosphatase B. And long story short, we decided to only focus on PAC1 because it had some very, very interesting functions when we look more closely into it. And we could confirm that PAC1 expression on a protein level in these islets treated with IGFBP7 was also reduced. It was reduced with around 15%. PAC1 supports mitochondrial function in beta cells. Interesting. And it supports mitochondrial function in numerous ways. It can regulate F-actin for insulin, for transport of insulin granules. It also is involved in regulating the electron transport chain. And in type 2 diabetes, PAC1 protein has been shown to be downregulated in type 2 diabetes, in protein lysates from type 2 diabetic donors. And there's also a beta cell specific inducible PAC1 which shows impaired, clearly implied in helping out the mitochondria in beta cells, this PAC1. So we wanted to assess how does the mitochondrial respiration work in our model system. So we performed the seahorse assay in the endoc beta cell line after treatment with IGFBP7. And here we see that after once again sent to our incubation with IGFBP7, we have reduced oxygen consumption rate after addition of pyruvate and after addition of FCCP. When we calculate from the ATP production, which is essentially the rate of change after we add a blocker for the ATP production step of the last step of the electron transport chain, we see that the ATP, the OCR dependent respiration, the drop here after oligomycin is also smaller in IGFBP7, showing that the ATP production is reduced after IGFBP7 treatment. So what we have is essentially less ATP production and putatively less insulin secretion. And we also have less max cells. So to summarize what we see IGFBP7 is fast insulin secretion, and it likely does this through reduction of PAC1 levels. However, we wanted to understand on a more cell type specific level how IGFBP7 is regulated in the islets, because so far we only have results in humans on whole islet level and in this beta cell line models, but we wanted to understand more what happens in alpha cells and beta cells respectively. So we looked into a small collaboration with, or not a collaboration, we just asked the people in our center who already had single cell and data published if we could also look at their single cell data. And here we see that the gene expression for IGFBP7 in alpha cells in type 2 diabetes is greater is greater. And this on a gene level, we don't really see this increase for IGFBP7 in beta cells in type 2 diabetes. But in alpha cells, it's a clear shift. It's a clear increase of the gene level of IGFBP7 in alpha cells in type 2 diabetes. And this is very interesting. But of course, mRNA is one thing, and then protein is another. So we hooked up with a really nice pancreatic from non-diabetic donors and type 2 diabetic donors, and stained them for glucagon, insulin, and IGFBP7. And then we used machine learning to identify islets and identify different cells in the islets, and then essentially look at the protein staining intensity of the intensity of the immunostain as a kind of crude measurement of protein levels for IGFBP7 in alpha cells. That in alpha cells the results matched, that we did have a slight increase in the protein content of IGFBP7 in alpha cells in type 2 diabetes, and also a slight increase in the protein content of IGFBP7 in beta cells in type 2 diabetes. So this was fantastic results. But as I've already alluded to, we wanted to look also at the crosstalk, the potential crosstalk capability, potential of IGFBP7 in pancreatic islets. So yeah, this I've already said. IGFBP7 is upregulated in alpha cells and in beta cells in type 2 diabetes. But we want to look at, like, is it in the same granules? Is it co-localizing with insulin and glucagon in the alpha cells and beta cells. So we perform the co-localization experiment where we have here Lunes. We saw, it's very clear in this image here, we saw that the Manders was greater than 0.7 for both alpha cells and beta cells in this dispersed islet stain, indicating that IGFBP7 is present in the hormone granules and is co-released with the primary islet hormones, which is very cool, very, very cool. So it's present in glucagon and insulin granules, suggesting that it is co-released. Lastly, in this paper, we wanted to manipulate this system. So we already treat the exogenous IGFBP7, but now we wanted to manipulate endogenous IGFBP7 levels in the cell line. And here we see that and for this we used siRNA for the IGFBP7 gene, and we have super clear knockdown on gene level. We have a reduction in the IGFBP7 protein content, and also importantly in this model here, we saw that the levels of secreted IGFBP7 into the medium of the was decreased. And while all this is happening, insulin secretion instead, so we have the opposite effect on insulin secretion. We lower the levels of IGFBP7 in this cell line. We also took this siRNA, treated 3 donors. These islet donors, it's like working with gold. When I defended my thesis last year, my opponent, he said, yeah, I mean, 3 donors here, but it's fantastic to perform in vitro, like your specific in vitro experiments on. And I couldn't agree more. Like, you're working with gold. And we're very grateful for the scientific infrastructure we have surrounding our islet research projects in Sweden. And in these 3 donors, we see that we reduce the mRNA levels after siRNA treatment, and we reduce the intracellular IGFBP7 protein levels, and concomitantly we increase, you could almost say that we improve the insulin secretion in these type 2 diabetic donors by 30%. So all of these results indicate that IGFBP7 in pancreatic islets can serve as a local autocrine negative inhibitor, stimulated insulin secretion from beta cells. So it's an autocrine inhibitor of insulin secretion from beta cells is our conclusion from all these results. And lastly, if we want to just quickly, but not extensively, but quickly just get an idea of the mechanism of action for this, and previous papers have shown that IGFBP7 can bind to the insulin receptor, it can bind to the insulin itself, and it can modulate insulin receptor signaling. Actually, what does what? We haven't figured out. We only know that IGFBP7 in the presence of increasing amounts of insulin on medium starved beta cells increases insulin receptor signaling, or receptor tyrosine kinase signaling in beta cells, and that we see with additions of increasing levels of insulin together with IGFBP7 here. So we have increased phosphorylation of it. And that is the effector protein of insulin and IGF-1 receptor signaling in beta cells. So we got like, we just dipped our toe a little bit in what this might be. And there are other papers already showing that insulin receptor signaling in beta cells likely is inhibitory, but that this is a little bit contentious in the field. But it likely is in most inhibitory insulins, which kind of makes like insulin can be an potent hormone, so it kind of makes from just cellular and physical secretion. To summarize what we have upregulation of IGFBP7 in both alpha cells and beta cells on a protein level, and we suggest that IGFBP7 is released. We know that it is released from beta cells. It is likely released from alpha cells as well. And we think that it works by binding or modulating insulin receptor signaling on beta cells and working on reducing PAC1 levels and thus reducing insulin, reducing clinical function and decreasing insulin secretion. And this also can have direct effects on calcium trafficking as well via the PAC1 here, and that's the pathway that we propose for how IGFBP7 reduces insulin secretion. And as I said, IGFBP7 is upregulated in both alpha cells and beta cells, diabetes, and when we reduce IGFBP7 in donors from with hyperglycemic states, we rescue insulin secretion. I went back there. So taking all of this into account, I have some thoughts and some future perspectives about what this project kind of means. We show that IGFBP7 is not regulated by blood glucose levels per se, but we know that type 2 diabetes is connected to low-grade chronic inflammation and a lot of other small stressful states in the body. Could it be connected? Could the regulation of this gene and this protein be connected to, for example, inflammatory state? IGFBP7 is upregulated by transforming growth factor beta 1, and this is very important in modulating macrophages and also in wound healing after inflammation. Could IGFBP7 have a potential role as an insulin sensitizer in tissue healing after inflammation? That is a question I've been asking myself. And if you have low-grade chronic inflammation in the islet, could that be the reason for the slight upregulation we have in type 2 diabetes? Unanswered question. And as I've mentioned before, inhibition of IGFBP7 reduces cardiac fibrosis, and it also improves, I forgot to add the nugget of information from this paper here. My mouse is dying a little bit. And improves blood glucose levels by reducing hepatic lipid content. So could targeting IGFBP7 be a multi-tissue drug target that helps in a lot of ways? Who knows? So that's where we kind of are in the big perspective of where I think research on IGFBP7 is right now. What it is already being used for, and I will be very brief on this, is that urinary IGFBP7 with another urinary marker called TIM2 have already been used as perioperative kidney damage markers. And IGFBP7-2 and TIM2 are believed to be secreted by stressed tubular cells and are more sensitive as early and subtle kidney damage markers as compared to serum creatinine and diuresis. And this nephro check, this is an already commercially available assay, although not widely used, it's commercially available. Actually shows really good results. It seems in a meta-analysis of how well NephroCheck performs, we see that measuring urinary IGFBP7 around 6 hours post-operatively can help you early, very, very quickly diagnose which patients might already be on their way to acute kidney injury post-operatively. And here we have like a really good area under the receiver operating curve, like with the performance of like 0.82 and 0.9 for assessing acute kidney injury. And these are really good results. This is a really, really good measurement for a binary outcome. So this is where IGFBP7 is already used. Although I must confess, I haven't seen it used in Sweden this way yet. Okay. With that, I would like to give my acknowledgement. This is my group. This is Lena, my professor, and Anna Maria, our technician. And back here we have Anna, who was my co-supervisor in my entire group, and LUDC, Lund University Diabetes Center, and also the collaboration with the cystic fibrosis groups we had internationally, and also our funding bodies. I also want to give a special shout out to Alexandros, who's our bioinformatician, who did a lot of the analysis that helped me here. And thank you all for listening. It's been a great honor to give this presentation to you. Thank you.
Well, thank you, Efraim. It was a very nice talk. I think that everybody enjoyed it. So I guess that we are going to go now to the questions that I guess the people have questions, I guess. So
Hola, Osvaldo.
Hola, Marcelo. I guess that you have some questions. Yes? Okay, well, since nobody is asking anything, I will start to break the ice. So, do you, I mean, this binding protein I think that is regulated basically through the DNA and transcriptional level, but also through processing. I mean, that is degradation. Do you look into that, anything of that? How, if it's up-regulated, down-regulated, what is you think that how the level of transcription is, the level of regulation take place? Some modification in the DNA?
Okay, so if I understand you correctly, you're asking how the levels of IGFBP7 might be regulated both
Right.
transcriptionally and post-transcriptionally as well. So I kind of we looked into what transcription factors might bind to the IGFBP7 gene, and it is as a supplementary table in the paper. And one of the highest, the top target was RUNX1, if I recall, and also PAX5. Our center did a really big study a few years ago where some of this data that I presented is compiled from, on how PAX is upregulated. And to answer your question, I don't have a clear answer on.
Okay. My second question.
Transcription factors and
Okay, okay, friend. My second question has
Sorry.
to do with my second question has to do with the levels of the IGFBP7 that you use in your experiments. Are they physiological? How, in terms of, I guess probably in the bloodstream, they are not as high. And so how do you justify the concentrations that you use?
So once again, a really good question. We did form a concentration determining or else with the consecutively increasing concentrations of IGFBP7 already.
I'm losing you. I can hear you.
Nanomolar, we started seeing the protein into the interstitial another cell. It could feasibly reach around 50 nanomolar.
Okay.
Osvaldo, there are two questions.
In the bloodstream, it's around 1.
Okay, I will pass the torch. I think that
It's significant. That much higher than what might be conceivably reached.
okay, we are having connection problems. Myself or everybody.
It's my
You see the questions in the chat,
Yes.
Osvaldo?
There's a question here from Marcelo.
Yeah, I'll open up.
He's asking, how does IGFBP7 regulates these cells and the release of glucagon and insulin under physiological and pathological conditions, such as type 2 diabetes? Did you get the question, Efraim?
Yeah, I can see it.
Okay.
So we show that IGFBP7 is released from beta cells. We don't show definitively that it is released from alpha cells. We're very certain that it does, but we haven't definitively shown that, just to be clear, or just to be honest. And how is IGFBP7? So on both, and the insulin receptor is expressed both on beta cells and on alpha cells. And insulin receptor of further insulin release, whereas insulin receptor signaling on alpha cells, so signaling relates it by increasing. In alpha cells, FBP7 would then increase insulin receptor signal and increase glucagon secretion. And you also see that a little bit in type diabetes, that you have hyperglucagonemia and that, like, hyperglucagonemia potentially driving high glucose levels, but it's such a big question here. I'm good. I hope you got something.
Yeah, the problem is that sometimes the connection is like unstable, so we
Yes,
lose
Can
you.
comes and goes.
Yeah, it comes and goes. That's right.
There's another question. Can you see it, the frame? It says,
I can see it from Andrea Milan.
yes.
Can you maybe wait 2 minutes?
Sure.
Yeah, sure.
And I can change my connection.
All right.
All right.
Yeah.
Yo le pregunté a Analía porque pensé que era yo sola.
No. Yo también tengo el mismo problema. Y fue empeorando, digamos.
Sí.
Is it better now?
Yeah.
Can you hear me clearly?
Yeah, it looks
Okay.
like, yeah.
Yes, right now.
Okay.
Yes.
We have a very ill-positioned router. I see the question from Andrea. Very good seminar, Efraim. Okay, I wanted to ask what function the INPP5B gene serves. They found it was differentially. Yeah, INPP5B can regulate essentially how different phospholipids in the plasma membrane work. So it could potentially change how the plasma membrane fluidity is regulated around the secretory complexes in beta cells. So it's very important that you have a very stable plasma membrane for efficient vesicle fusion. And if you have a plasma membrane that's a bit too fluid and unstable, that could potentially lead to inefficient insulin secretion. That's one of the theories we have around how that gene might be involved. But we simply chose to do our experiments on PAC1 because there was already a lot of research done on PAC1, and it seemed the most appropriate candidate to follow up for us.
Okay. More questions?
Any more questions?
Well, I have
a question. I have a question.
Okay, good.
You said that the inhibition of IGFBP7 reduced cardiac fibrosis, right?
Yeah.
Was it reported in diabetic patients or just in patients with cardiac fibrosis from any other cause?
So in that paper, the experimental, when the manipulation of IGFBP7 was only performed in mouse models.
Okay.
They manipulated in mouse models for heart failure. And they manipulated IGFBP7 both by suppressing the gene expression in cardiac tissue and also by adding anti-IGFBP7 antibodies. And they saw that doing both things rescued heart failure or improved heart failure related outcomes in these mice. The patient data from that paper show that in heart failure, you also have increased serum levels of circulating IGFBP7, but they don't perform any manipulation of IGFBP7 levels in patients. They only descriptively look at IGFBP7 in heart failure patients there. So they don't give the antibody, for example, to the patient with heart failure.
So the origin is not known. I mean, they see that it is increased, but they don't know where it comes from, or it comes from the heart.
In that paper, they theorized that it's secreted by fibroblasts in the myocardium. But what they saw, they could regulate it both directly in cardiac tissue with, I think it's adenovirus vectors that are taken up by cardiac tissue, and then the level of fibrosis is decreased, but they also gave antibodies to bind IGFBP7, and that also decreased the level of cardiac fibrosis.
Okay, thank you, and a very interesting lecture. So thank you very much for accepting this invitation.
Thank you very much.
Give us.
I have one more question, as just a curiosity. Do you show colocalization of insulin and this growth factor, right? Do you think that the interaction take place at the Golgi level or some other stage?
I don't. I'm actually pretty sure that if IGFBP7 is present in the insulin granules, yes, but the way insulin is stored in the granules themselves, insulin is not stored as the free peptide, because that would conglomerate. It's actually stored in stable hexamers that are then dissolved upon release. So the way the insulin is stored and folded unto itself in the insulin vesicle makes it unavailable for IGFBP7 to bind within the insulin vesicle. I think that is one of my favorite little factoids about insulin vesicles, that I don't think a lot of people know, that insulin is actually stored as a hexamer together with zinc. It needs a zinc ion for this hexamer to form. There's some great papers on that.
Well, is there any more questions around? It seems like not. Oh, okay, well, Efraim, thank you very much for the presentation. I think that everybody enjoyed us as much as I did. And we hope to see you when you visit Argentina, Buenos Aires, and just show up at the Institute. Maybe if you have some new results, you can give a short talk. I mean, I saw that you have several papers, some of them that you didn't present any data today. So maybe one of those can be a nice topic for a presentation, if you feel like.
Yeah, I mean, we have one out for review in Diabetologia right now. Maybe that one on cystic fibrosis. Who knows where we are with that one? But I can't show that one today because it's completely unpublished.
All right.
But maybe, who knows?
Well, good. Well, thank you again. Hope to see you in Buenos Aires. Okay. Thank you again.
Okay.
Reciprocal.
Thank you.
Reciprocal. Thank you.
Okay. Goodbye.
Bye.
Bye.
Bye. Bye. Hope to see you